This is Dr. Launen from Keene State College, and this is the first of two mini lectures looking at eukaryotic genomes from a very general perspective. In these two mini lectures, we're going to consider the basic physical structure of eukaryotic genomes, the fact that they're linear, what it means to be diploid versus haploid, and the terms chromosome and chromatin, what they really mean. We're going to consider the variety of sizes in eukaryotic genomes briefly. We're going to talk just a little bit about the role of repetitive DNA in eukaryotic genomes and the idea of gene count and how much it does or doesn't matter. And we're going to talk about genome compaction. And then we're also going to talk about how the state of chromatin affects gene regulation in eukaryotes. So eukaryotic genomes, by definition, are found, or at least the nuclear genome is found in the nucleus. So here's a figure taken from Khan Academy of, say, an animal cell. There's no cell wall, so I think it's an animal cell. And this big purple structure is the nucleus. So the nuclear genome is in the nucleus, and we'll talk more about all these bits and pieces in a moment. But that would be where in humans our 23 pairs of chromosomes would be located. Now there's more to the genome than just the nuclear genome in a eukaryotic cell, because eukaryotic cells have additional organelles. All eukaryotic cells, or almost all of them, have mitochondria for the purposes of generating ATP through respiration. And mitochondria are shown here, here, and here. And each of those mitochondria contain their own small circular genome called the mitochondrial genome. Now, not shown in this image because it is an animal and not a plant cell is a chloroplast, but chloroplasts are the small organelles that are responsible for photosynthesis, and they also have a small circular genome. So when you're talking about just the eukaryotic genome, you would be best off describing that as all of the genetic material within a eukaryotic organism. And then maybe being a little bit more specific and talking about whether you mean the nuclear genome, the mitochondrial genome, or the chloroplast genome. So we're going to talk about mitochondrial and chloroplast genomes in another lecture, but I do want to remind you of where those particular organelles came from in the history or the evolution of eukaryotic cells on Earth. And that is to remind you of a theory called the Endosymbiosis theory. The idea that eukaryotic cells gained most of their organelles, specifically though the mitochondria and the chloroplast, through a process called endosymbiosis. So one cell phagocytosed another, and the one that was phagocytosed persisted and became an organelle, which explains the double membrane structure found in mitochondria and chloroplasts, and also the fact that when you look at their mitochondrial DNA, what you find is it's circular and it contains genes that have prokaryotic-like structure. They don't have introns. The 16S RNA gene is present, and so on. And we'll talk more about these genomes in a subsequent lecture, as I said. So here's a figure from the free open source biology textbook just called OpenStax Biology, and it's purple again. And so here's the nucleus. And the nucleus has a lot of really interesting structural features. So it has a double membrane. So it has two layers of phospholipid bilayer. It has an inner one, and then it has an outer one. And that outer one is continuous with the structure called the endoplasmic reticulum, which is this sort of net of membranes shown here. We won't talk much more about the endoplasmic reticulum. Inside the nucleus is structure that is referred to as nucleoplasm. So kind of like the cytoplasm, it's a liquid with a lot of electrolytes and a careful pH balance. And then there's a lot of spaghetti-like material, and we'll get to that soon, and it is called chromatin, and that is where the DNA is found. So note here, and this is a preview to what I'll be saying more of in a moment, you don't see any specific lines or X-like structures when you're looking at this cross-section of a nucleus. And that's because most of the time those structures that we refer to as chromosomes cannot be visualized. Instead, we see a spaghetti-like mass known as chromatin that has all of the nuclear genome sort of compacted within it. When you're looking at a nucleus, there's always a dark circular area called the nucleolus. And that is a particular area that contains DNA that has ribosomal RNA genes on it. And this is also the place where ribosomal subunits are assembled. So you should remember that ribosomal subunits are made out of rRNA and made out of ribosomal protein. So the rRNA is made by transcribing the genes that are in the DNA in this region, and then the ribosomal protein is made somewhere out here by ribosomes, and then it's actually sent back into the nucleus through nuclear pores, these structures here, and then all of that is assembled here into ribosomal subunits, and they are exported out of the nuclear pores to form these little ribosomes shown as little red balls here. And that area always stains really dark microscopically so we can see where it is quite distinctly. When talking about eukaryotic genomes, it's important to remember the definition of the terms haploid and diploid. These are adjectives. So to be haploid means having one complete set of chromosomes, and to be diploid means having two complete sets of chromosomes. In other words, each chromosome is present as a pair. Whether they're lined up together or not, they're there in the nucleus as a pair. And these terms, of course, are relevant only to eukaryotic genomes because prokaryotes, by definition, are permanently haploid. In eukaryotes, the gametes are haploid, and the somatic cells are diploid. So let's think a little bit about genome size and how it might range. And we've looked at this figure before as we considered prokaryotic genomes. And so I'll point out to you again that this is a logged scale, and that this figure shows haploid genome size, measuring the nucleotide pairs that are found in the haploid genome, and that allows us to compare eukaryotic genomes, which is everything here from mammals down to protozoa. They're all eukaryotes. It allows us to compare their genome size with prokaryotic genomes because you're comparing apples to apples, haploid to haploid. So here's the typical range for mammals, and that's where humans fall. In the billions range, 10 to the 9 is a billion, of course. The plants tend to have much larger, more complex genomes, and so do a lot of the amphibians, the fishes, crustaceans, and so on. And so I want to make the point as we're looking here that, if you hadn't actually studied genomes, you might assume that, humans are the most complex organisms on Earth, and I think you can make a pretty good case that that is so. And so you would expect that, maybe we might have the biggest genomes, but in fact, genetic studies have clearly shown us that that's not the case, and that there's no linear, positively correlating relationship between genome size and organism complexity, or indeed between gene count and organism complexity, that it's a lot more complicated than that. You've looked at this figure before as well in an earlier lecture about prokaryotic genomes, or you will be looking at that lecture about prokaryotic genomes soon. So this gives us some numbers that haven't been logged. They're maybe a little bit easier to look at, and it also gives us a gene count. So here we are, humans or Homo sapien. We have a genome that's about 3.2 billion nucleotides in size, about 25,000 genes, and that means we have an average gene density of about 1 gene per 100,000 bases. Here's the mouse, very similar density, actually rather similar genome size. The fruit fly's got a much tinier genome and a more compact gene density. And then if we go down here and we look at the two representative prokaryotes, E. coli and Haemophilus influenzae, and we see they have a very, very dense gene count per number of bases. So prokaryotic genomes are highly efficient, and most of the DNA is organized into genes. Eukaryotic genomes, in contrast, tend to have relatively low gene count for the total amount of DNA present. So what they have is a lot of extra DNA that is not genes. It's things like repetitive DNA sequence, and we'll look at some of that in a moment, but it's not actually coding DNA. Eukaryotic genomes have an abundance of non-coding DNA, not DNA that is meant to make a functional RNA molecule, not DNA that is meant to make a protein or peptide more accurately. All right, so let's consider 3 definitions here. Because I want to talk about the term chromosome and chromatin next. So DNA is what? It's a nucleic acid. There are 2 nucleic acids, DNA and RNA. Okay, deoxyribonucleic acid. Na is for nucleic acid. One of the categories of macromolecules that you should know about. it is always found as two complementary and antiparallel strands that form a double helix. At least this is true in cellular genomes. And it contains the genetic information. We already know a lot about DNA and how that flow of genetic information works. So what are chromosomes? And here I'm going to confess that, like a lot of people who tend to focus on organisms from a very molecular perspective, I often use DNA and chromosomes somewhat interchangeably. And that's not actually good technical practice because chromosomes are actually somewhat different. Chromosomes are DNA molecules complexed with protein and RNA that are only visible during cell division. So by their strictest definitions, chromosomes don't actually exist in the nucleus all the time. DNA molecules do, but the DNA exists most of the time in the nucleus in a structure that we'll call chromatin, this big spaghetti-like structure that is DNA, protein, and RNA all complexed together, and it includes all the DNA molecules, the entire nuclear genome. So in the next lecture, we will continue considering, the next mini lecture, we'll continue thinking about these topics.